Keep on.
We have some people have to do too.
Battery full छैन? किन गर्छस् साले? Estamos de acuerdo. खेल्छु तिमी गएर आउँछ
Hi friends, hi all done. Today I have got the topic of electrophoresis. Basically we did on our last class, previous last class, was the electrophoresis of the protein. You see how the protein moves to their respective electrodes with their respective charge on them. But I'm here talking about the cell electrophoresis. Let me introduce my topic first. Our topic is about the cell separation with the horizontal cell electrophoresis under isopycnic conditions on a density cushion. Isopycnic conditions, you all know about isobaric thing, right? That's the same pressure. Isothermal, that's the same temperature. And this isopycnic thing is the conditions when there's the same density provided. Let me first introduce a little bit about the cell electrophoresis itself. It's a technique that permits identification and measurement of the electrochemical properties of the cell surface. We all know that the cell wall, cell membrane have various cytoskeletons and various proteins like glycocalyx, glycoproteins that represent the electrochemical properties of the cell. And this cell electrophoresis application is applicable to those changes that occurs during cell division or cell differentiation. So we basically take the samples of those cells which are rapidly dividing and rapidly differentiating. The horizontal electrophoresis systems is effectively separated human and chicken RBCs, red blood cells, that just differ 30% on its electrophoretic mobilities, carried out under isopycnic conditions when all the cell sedimentation was considered negligible. The medium used for the cell electrophoresis here was an anti-convective porous matrix of polyurethane sponge. It's the polymers of the urea and the ethane itself, with a pore size over 300 micrometer. Basically, I was trying to compare the two cell electrophoresis here. One with the use of polyurethane matrix, it's PU. The short form is PU, and other with the solution 2, that's a 9 gram dextran, 50-20 TDA, 7 gram sucrose in 100 ml of 90% water and 10% 0.9% NaCl. The outcome was quite interesting because the outcome was quite interesting because you could see the electrophoretic movement of the cells comparing between the two solutions used. This is the diagram actually that shows the cell electrophoresis of RBC and EOL1, that's the eosinophil 1, that's a leukocyte type of leukocyte cell. On the first medium, the picture A shows the cells before the electrophoresis. In the picture B shows the cells after the electrophoresis. On the first on the first diagram, the RBC has shifted its place after the electrophoresis was done, and the eosinophil 1 didn't shift its place because eosinophil might have binded to the polyurethane matrix that was used for the electrophoresis of the first sample. And on the second sample, solution 2, what I was talking before, it actually does move. This is the cell electrophoresis of the human and chicken RBC. The third kind is a mixture of RBC, that's both of chicken and human. It's done on the solution 2. You can easily see the RBCs of all the RBCs that have been shifted after the cell electrophoresis. And also the RBC and the eosinophil 1, the eosinophil that didn't move on the cell electrophoresis on the first solution, it actually didn't move on the cell electrophoresis on the second solution. So basically overall the whole experiment was considered quite beneficial for the characterization of the electrophoretic mobilities of the different cells between different species and the different cells between the same species. By this way we can easily, by this process, it was quite like in our modern biotechnological research is going on. This experiment was quite helpful in differentiation of cells of different types. Thank you.
Next, and then Ram after Nisha.